nervous system, and we're first going to start with the evolutionary history of the set nervous system. So the simplest organism with a nervous system is a cnidarian, which is the same as a hydra, and they had a nerve net, which is basically a loosely organized system of nerves, which is bidirectional impulses, which means that throughout the hydra, the impulses can go forward, backwards, up, down, in any direction. And stimulation at any point, it spreads across to cause movements. If you touch it here, the whole thing would move. And the primary function of the... an example of evolutionary history for nervous system could be cephalization, which is basically the concentration of sensory organism at the head, the formation of the head. As time went on, more complex organisms began to develop these heads, all these sensory organs. And
Alright, so for invertebrates, the nervous system is a little bit different than vertebrates. For the flatworms, they are more complex than the hydra, but less complex than, let's say, octopus or a cephalopod. They have a diffuse ladder-like nervous system. You see here, their neurons are sort of aligned in like a straight line, causing more variation in the movement and the motor senses of the flatworm, so they can move different parts and not have to move their whole entire body. This is very evolutionarily important because we can start to see how perhaps the spinal column came about due to the centralization of the nerves in the flatworms. But for cephalopods, they're more complex because they start to develop a brain and a head. For example, the octopus, it has a very large brain, large eyes, and large axons, meaning that it can send its impulses on its nerves a lot faster and a lot stronger to do more complex things. For insects, though, they have a giant fiber system which allows rapid conduction of nerve impulses, so they can do a lot of things in a very short time and send lots of their nerve impulses. The early brain that we would see was divided into different parts: the protocerebellum, the duocerebellum, or cerebrum, and the tetocerebrum. And they're sensitive to odors, sounds, and lights compared.
Alright, these are the parts of a nervous system. We have the central nervous system, the CNS, and the peripheral nervous system, the PNS. Basically, the central nervous system is your brain and spinal cord, and it's the main part of our nervous system. The peripheral nervous system includes our sensory neurons and motor neurons. We'll go over those in just a second. And the peripheral nervous system is also known as the autonomic nervous system, which break down the sympathetic and parasympathetic. The sympathetic response is basically the flight or fight response, is when you have an adrenaline rush, your body tenses up, your heart rate increases, and your digestive system slows down. Then you have your parasympathetic, which is your at-rest response. Your body relaxes, your heart rate decreases, and your digestive rate increases.
Alright, so now we're going to talk more about how neurons work. So, here's a neuron. There's a dendrite sticking out of it. And it's a normal cell inside with a nucleus and everything. And they have these things called axons, which are like long tubules. And along the axon we have the myelin sheaths. And these are useful because the charges travel along these myelinated sheaths, and they're good conductors of electricity. And all the neurons are interconnected to other neurons. And you have the motor neurons, and you have the sensory neurons, and you also have the... Okay, so we have two things called ISPS and ESPS. So what ISPS does is it inhibits the channel, so it makes it so that they don't work. But we have the ESPS, which excites it, so they do work. So a good way to remember is excites, so yeah, and ISPS, so inhibits. And in real life, we have an example of like an actual way this channel works. So say you're pricked on your finger, just like pricked. Your sensory neurons will pick up the pain, and it will send the message through your interneurons all the way to your CNS. So it'll go down to your spinal cord, it goes to your brain, and then your brain will receive that signal, and then it sends a signal back to all the way down to your motor neurons, which will then move and cause your hand to move away.
As the action potential travels down the axon through the neuron, it reaches this thing called the synaptic cleft, which is basically the space between these two neurons. And so what happens is the action potential is traveling down, and there's this thing called neurotransmitters inside vesicles. Neurotransmitters are things that are the signals that are passed, chemical signal. Example would be like dopamine. So what happens is calcium ion channels, they open, and calcium flows in, which causes this vesicle filled with neurotransmitters. There's more than one. Neurotransmitter vesicles. And they travel down. They travel down and they bond with this and with the membrane, and they release their neurotransmitters into the synapse. This gap right here is called the synapse. And so, as these neurotransmitters are making their way across the synaptic cleft, the synapse, there are receptors on the dendrites of the other neuron, and these neurotransmitters, they will bind to these receptors and would then trigger another action potential to be carried on through to carry on the movement. So if it's ISPS reaction, this action potential would not occur, and it would stop before it ever reached the actual other neuron. If it was an ESPS, it would be stimulated even further to make more of the reaction. And so as the calcium flows in, the vesicle releases the dopamine, or the neurotransmitter, across the synaptic cleft to the receptors here. And afterward, when it's done, enzymes come in, and it clears up all these leftover neurotransmitters in the synapse, and they're recycled back into the vesicles to be used later on. Speaking of ISPS, there are other things called alcohol and drugs that can inhibit the response. So these are represented by triangles. The drugs enter your system and they mess with your response system. So certain drugs and alcohol, they come in, they bind to the same receptors, and they release the neurotransmitters. And instead, these bind, which can either excite your neurons beyond capacity or make them dead and not have any reaction at all. This is why alcohol and drugs is dangerous to your system. Don't do drugs.
Alright, so action potential and saltatory conduction. If you look at the neuron, it has a lot of potassium on the inside and a lot of sodium on the outside of the neuron. But you might think if it has a lot of potassium and the potassium ions are positive, you would think that the inside of a neuron is positive. But actually, the inside of a neuron is negative. It's about negative 70 milli. So when no action potential is going through a neuron, it starts at negative 70 millivolts because of all the negative proteins and chlorine atoms and stuff that are actually inside of this neuron. And if you look at a neuron, it also has a period that it's covered up. It's covered by a Schwann cell, which produces like a sheath called the myelin sheath. And at this point, there's no action potential because there's no gates. And to talk about the gates, there are sodium gates on the membrane of a neuron. And when a signal is being passed and the neuron gets excited, the sodium gates are opened up. And as you can see here, I drew arrows showing all the sodium ions going into the neuron. And that makes the graph spike up, becomes really positive as more sodium rushes inside the neuron. And that was what makes the signal. And after a while, that triggers potassium gates to open up, and then the potassium is going to exit the neuron. And after, you see that it kind of overshoots. So all the potassium is leaving, and too much potassium leaves, and you sort of dip down. But right here we have a sodium and potassium pump where both sodium and potassium can come in and out, resetting this resting potential. And what happens is, in a space where it's covered up by the myelin, this signal sort of dies down, but not to a point where it's not positive enough. So when it's not covered by a myelin, the next space, this voltage is still positive enough to trigger another action potential. And what this does is it makes it really, really fast, sending multiple signals at one time down the axon to travel a great distance at high speeds. Amen.
What are the main parts of a neuron? If you said cell, dendrites, axon, and synapse, you are correct.
What type of nervous system do hydras have? If you said nerve net, you are correct.
Which invertebrate has the most sophisticated nervous system? If you said cephalopods, you are correct. What is the difference between ESPS and ISPS? ISPS is an inhibitory response for a neuron. ESPS is the excitatory response, passing on the signal.
What is the difference between sympathetic and parasympathetic response? Sympathetic response is a fight or flight reaction, while a parasympathetic response is when your body is at rest. What is the charge of a neuron at rest? Negative 70 millivolts.
What is saltatory conduction? It is when the impulse jumps along the myelin sheath.
So we have ISPs and ESPs.
ISPS. No, keep going. Keep going.
You can just start over.
No, we're still going.
No, we're not.
It'll be funny. Come on.
No, it's not. What is the charge of the neuron when it is? What is that?